questions before we get rolling? Shiny. We recording, my dear? Thank you. So on Monday, we had just finished off starting our literal tour through the cell. And so remember, as we are doing this, we're kind of starting inside out. Okay, so we started and had just finished talking about the nucleus and that nuclear envelope, right? So we talked about DNA being our books in the library, and the nuclear envelope being the library walls. Excuse me, keeping all of that safe and protected. And so from there, right, we're going to move our way along, okay, through the cell and figure out, well, what's happening at each sort of place in the cell. Okay, so we started at the nucleus. Okay. And its nuclear envelope. Okay, and we know that DNA is there. That's what we keep safe. Okay, so now we want to think to our biomolecules a little bit. Does DNA leave the nucleus? What leaves the nucleus? RNA, good. Okay, and we're ready to leave the nucleus. So now we're going to say, if I want to do something, if I want to make something, where can I go? So let's try to make some stuff. Okay, so if I want to make something, I have several options, and it depends on what I want to make. Okay, so the first place I can go is the ribosome. Okay, and ribosome is going to be one of our top stops because it makes really important stuff. Okay, as you can see, it makes our favorite biomolecule. Okay, so we can make proteins at a ribosome. Feels like a big deal. Okay, so there are two types of ribosomes in our cell. Okay, what we call free ribosomes. Okay, literally meaning that they're free floating or they're not stuck onto anything. Okay, so they're floating in what's called the cytosol or the cytoplasm. Okay, so remember, here's that prefix meaning cell. Okay, so we've got ribosomes just living their best life in the ocean of the cytoplasm. That's what we see here. Living their best lives. Okay. The other place we see ribosomes is bound, literally, to an endoplasmic reticulum, okay, on the rough. And we're going to compare this in the next slide. Okay, so that endoplasmic reticulum becomes rough because these ribosomes are on it. Right? So we get all bumply. So we can see all the bumples here. That's the technical term, bumples. Okay, so the key here, regardless of where we go, a free ribosome or the rough endoplasmic reticulum, right, with the bound, if we see ribosome, it's something protein. Right, we're going to make a protein at a ribosome. That's its core job. Keep track of our journey, going across the top until I run out of space. Okay, feel good so far? So let's look at, turn this off, I can't erase the whole screen anymore. Our endoplasmic reticulums. Okay, so let's just focus on the left-hand side of the screen first. Okay, so here's that rough ER we just talked about on the last screen. Okay, so remember our rough ER has bound ribosomes on it. Okay, and so remember, if we have ribosomes, okay, we're going to remember ribosomes are something proteins. That's going to be the easiest way to remember this. And anything that has to do with the ribosome, okay, details aside, I'm going to make a protein. And so if I look at my image down here, beep, 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 beep. Split that in half, okay? The right hand of that image down there is showing my rough ER, right? And I can see again all of those bumplies. I'm sharing my screen. Oh, gracious. So again, we can kind of track all those little bumps. Remember, those bumps are the bound ribosomes. Okay, so as we can see from our list, the rough endoplasmic reticulum makes a lot of stuff, right? Glycoproteins. Okay, remember glyco, we've seen this before when we did biomolecules. Glyco is like glucose. Actually, this is the combination of a protein and a carb. Okay, but right, something protein. Okay, this also has to do with transport vesicles, which we've seen in lab. Okay, but key here is something protein. Okay, this is the same way that you'd see it on the test. Okay, if I'm talking about a ribosome or a rough ER, okay, you're going to see something explicit about proteins for the right answer. Okay, let's compare that to the smooth endoplasmic reticulum, right? So if we look down on my left-hand side of my image, where my smooth endoplasmic reticulum is, the thing that makes this smooth is the fact that it doesn't have those bumplies, right? And those bumplies are my ribosomes, right? So being ribosome-free, all I have is this membrane. This is iconic for eukaryotic organisms, right? We have membrane-bound organelles. And in this case, all I am is a membrane. Okay, but if I don't have ribosomes, I don't make proteins. That's my big key here. And so as we look at the list of what the smooth ER does, I see that it does a lot of stuff. In fact, I see a lot of familiar stuff, right? Look, lipids, that's a biomolecule I recognize, right? Carbohydrates, that's another biomolecule I recognize, okay? So this pretty much does everything but. Right? We can't do proteins, right? No ribosome, no proteins. So we can add a second track here if we want to. I don't know how long it's going to keep it up, but I'm going to lazy hand that. So I'm going to go to some kind of ribosome place. Oh, it's super cut off at the top. If I want to make a protein, I'm going to go to the smooth ER if I want to make some kind of other biomolecule, like a lipid or a carb. Okay, this feel okay so far? Okay, any questions about either of these? Rude. Oh, I knew I missed one. Okay, so what if instead of making something, right, so that was our journey to make something, what if I needed to break something down? Okay, so I have a really big compound, right? I just ate a cheeseburger. Okay, you can't do anything with the cheeseburger itself. Okay, you have to do stuff with the bits of the cheeseburger, right? We absorb small parts of the cheeseburger. How would we do that? So let's take a look at that. Okay. The main way that we just stop, break things down is via the lysosome. Which is sort of like a stomach. Okay. So our lysosome is basically a sac, okay, of hydrolytic enzymes. So here's how we can kind of remember this. We've seen this prefix before. Remember we did hydrolysis, right? So we've seen lysis. Remember, lyse is to break. Okay, so that's the same thing here. Lysosome, lyse, to break. Okay, so like our stomach is functionally just a sac of acid, right, that breaks stuff down. A lysosome is also just a sac, right, in this case, of hydrolytic enzymes. Okay, so again, lytic, to break, and hydro meaning, how are we breaking this down? With water. Okay, so enzymes that are going to break things down with water. Autocorrect, I ain't got no patience for you. Even when I make slides, I can't spell. Okay, so the key here when we look at this is this functions the same way your stomach does, right? When you eat a cheeseburger, right, that drops down into your stomach, okay? And the goal of your stomach is to break that down into teeny weeny bits that you can use. Okay? Our main goal here as we look at this, and these first two bullets is, here we have a sac in the cell whose main goal it is to take large chunks, okay, from a cellular standpoint, and break them into smaller bits. Okay? So a big cheeseburger for them is a macromolecule, right? Macro means large. Okay, so we're taking larger proteins or fats. Remember a polysaccharide? Poly means many. Okay, so we're basically taking large chunks of things and breaking them down into smaller pieces. Okay, so quite literally, the version of the cellular food and nutrients, breaking them down into usable pieces. Right, the other thing that we see, which is this bullet here, is some recycling. Okay, so one thing we're finding is that your cells are much better at sustainability than people are. So anything that's going bad, okay, so a mitochondria that's been around a long time and isn't doing a very good job, for example, of making energy anymore, okay, that would maybe go to a lysosome. Okay, we're going to tear down, break down that mitochondria. Okay, but we're not just going to throw it away. Tisk, tisk, you're not doing your job. You're going to the mitochondria junkyard. All right, so there are any junkyards in your cells. Okay, if something like that, when everything breaks down, okay, when that happens, we're going to shove it into a lysosome and we'll break it down into its parts and then use those parts to make new things, maybe a new mitochondria, okay, maybe a new cell membrane, whatever the case is, right? Just because the mitochondria isn't working anymore does not mean it doesn't have parts, right, carbohydrates, lipids, whatever it is, that are not still valuable. Okay, so reduce, reuse, recycle, right? Right, so that's the job of the lysosome. It's not actually going to be doing the reusing. It's just breaking it down and telling the cell, hey, I have these parts, you can come get them. All right, so our lysosome or our cell stomach making sense. Okay, so let's start there. If my lysosome broke down something, okay, what would I do with that then? Where would I go from there? Okay, so I just said that the lysosome itself isn't going to send those bits to wherever they need to go. So who does? Whose job is that? Ooh, that's absolutely terrible. Let's see if I can shift that down a second. I knew that was bound to happen. Okay, so no matter who's done something, so we could be coming from the smoothie R, right? When I finish something, okay, they're not sending it anywhere. Okay, if I break something, they're not sending it anywhere. Okay, there's one person whose job it is to do all of the shipping, and that is our Golgi body or Golgi apparatus. Okay, so this is Amazon, the Amazon shipping center, or UPS, or whatever it is that works for you here. Okay, so as we think about this, okay, this is the center in the cell that gets all of the shopping requests, the shipping requests, okay? So if there's a part of the cell that says, we need more nutrients, right? Or we don't have enough barrier wall over here, right? I just ordered a brand new box of cell membranes. 2-day shipping. Okay. So we already know if I'm going to make a cell membrane, that's a lipid, right? That's going to be made from my smoothie R. Okay, so we've got different places that are distributing, but they're all going to come to the same shipping center. Everybody's got to go through Amazon. Ain't it the truth these days? All right. So our Golgi apparatus has all the shipping orders for the entire cell. So everything we make, everything we break has to come here to get bagged and tagged. So here's how our Amazon shipping center works. So let's take a peek at the image that I have here. So our Amazon shipping center, our Golgi body, has a very efficient warehouse system. So we have one side, okay, where all of the distribution orders come in. Okay, that's our receiving end, or what's called the sys face. I don't think yellow is going to look very good here. Okay, and that's our convex side. right? Or this rounded edge that we have here. So we can see this is outwardly rounded. So everything's going to come in on that edge. Now once we get here, we see that everything is shaped in what we call cisternae. Okay, so we're membrane-bound organelles, right? It's a eukaryotic thing. So ultimately what this looks like is if you kind of put a little bit of air in a bunch of Ziploc bags, right, and then stack them on top of each other. That's what our cisternae is. So here we have all of our Ziploc bags, right, filled with a little bit of air, all stacked on top of each other. Okay, making our cellular warehouse. Right, our one direction warehouse. So everybody's going to come in on the cis face, and as they travel through our cisternae, several things are going to happen to them. Okay, so our products, say our lipids that we've brought in, okay, are going to get unpackaged from however they've come in, right, unboxed, for example. Okay, they'll get checked, sorted, and repackaged. Just like you'd imagine would happen at a real Amazon shipping center. Okay, we're going to scan and barcode them, see where they're supposed to go, rebox them up. Okay, when we rebox something, they're going into what's called a transport vesicle. Okay, it's like a little car, right, which is also made of the same thing that our cell membrane is made out of. What are cell membranes made out of? Phospholipids. Perfect. That way, if it needs to leave and go through the cell membrane, it can, because they're all made out of the same stuff. And then when everything's ready, when it's been boxed and barcoded, okay, and ready to go, then it's going out the opposite end. Right? What we see is the concave side, which is the side that looks like a loop or a C. This is the trans face, okay, or the shipping side. Okay. So as long as everything's going unidirectionally, nothing gets bumped up, right? Nothing smashes into each other. And it says modified products, like how so? Like what? So the question is, it says modifies products. What can that mean? So in this case, we can combine some things, right? So you can combine proteins and carbs together. You can also lengthen and shorten them depending on what the order is. Okay, but when we say modify, right, we're not creating from scratch, right? We would just slightly alter in like very small ways, so like shrink or combine. Or maybe cap, right? Okay. So whether we're breaking something or making something, whatever comes out of what we've done, everything has to go to our Golgi body. Okay. Any questions about our Golgi body? Pardon the story up top. Okay, whoop, yeehaw. Okay, so this is our story so far. All right, we started in the nucleus, that purple bit in the very center. Okay, from there, we were able to go either to the smooth ER, the rough ER, or some free ribosomes. For example, if we went to, say, the smooth ER, that would be one example of a direction. From there, we would go to the cis Golgi, through to the trans Golgi, right into a transport vesicle, where we would be shipped out to wherever Amazon said we needed to go. And that would be dependent on the needs of the cell, right? Where is that lipid need? So here, for example, we're putting our lipids into the plasma membrane. Okay, so this would be a good way to practice this. Okay, work yourself through some pathways. That's the one I want? Yes. Okay, so let's practice some mentees. Okay, remember, we're not going to use our notes while we're doing this, right? We're practicing studying. So we're going to go to menti.com, right? You're going to enter the code 76 10 18 9. Okay, and go through and not only decide what the best choice is here, but spend some time thinking what exactly, right? Do this like we would do in lab. What are the wrong answers do? All right, let's start at our least popular answers and work our way up. I'm writing, okay. So let's look at lysosome first. So if you did not choose lysosome, why did you not choose lysosome? What did you say lysosome did? Breaking things down. Good. Is that a question or are they answering? They're answering. Good. Good. So our lysosome, remember, we're going to think lys, to break. Okay, so remember, this is our stomach. Good. All right. Golgi body. This is my favorite organelle. Gotta have a favorite organelle, right? So our Golgi body, if you did not choose Golgi body as your option, Why did you not choose Golgi body? What did you say it did? It's a nice looking P right there. Packaging and transport. Good. Okay, so it's not making anything itself, right? It may alter things, but it's not going to make them, right? Good. All right, let's hop over here. Okay, so we have just the ERs left, so some kind of endoplasmic reticulum is likely our choice. Okay, we know that we need to make something. Both the ERs do that. Our goal here is to make a lipid. So if you did not choose the rough ER, why? You can hear voices, but they're not quite carrying through the soundproof ceiling. It makes proteins. Good. And how did I know that? Has ribosomes, right? Rough is ribosomes. And anything with ribosomes, right, is something proteins. All right, good. Soothe ER, right? Really having a day with this, aren't I, guys? All right, so my soothe ER, right, or smooth ER. All right, so we know this is the answer by process of elimination. Okay, but I do want to do better than that. So why do I know that the smooth ER is the right choice? Because it makes everything but proteins. And we know this because it has no ribosomes, right? It's smooth. It's smooth because it lacks those ribosomes. Okay, so if this were a test question, this would be how I would break this down, how I would work through this. All right, great job, everybody. Okay, any questions about that before we move on? All right. Okay, so thinking about ways to study, right? The way that we did this in the before we did our Menti is a really good way to do it, right? So think about kind of doing a little fake tour, right? Take a starting point and work your way through from a start to finish. It makes a really effective study method. It's a little more fun than just sort of like stock writing out definitions. And anytime that you're not totally bored when you're doing it, helps engage your brain a little bit too. Okay, any questions so far? All right, so let's look at now, okay, up to this stage we've been talking about how do we move around the cell. So now we want to talk about how do we have a not dead cell? Okay, how do we keep our cell alive and kicking? So one of the first things we're going to look at is energy for our cell. So there are several organelles that help create energy. Any given cell, depending on what they are, may have one or more of these. Okay, so we're going to go into these first two in a bit more detail. They're more common and more efficient at how they create energy. Okay, we're not going to talk any more about this last one beyond this slide. Okay, it's less common and it's also significantly less efficient at what it does. All right, so let's talk first about the mitochondria, the powerhouse of the cell. Okay, probably all at least somewhat familiar with this one, if not for the memes. Okay, so we know mitochondria is one of the most efficient ways that we make energy. Okay, we call it cellular respiration. We'll have a whole talk about this later on. Okay, we also call this metabolism. Okay, the ability to use an oxygen-rich environment and convert sugars or some kind of energy-rich fuel into ATP. Okay, we're going to talk a lot about ATP. We've already mentioned it in lab if you've had that with me this week. But ATP, I almost did it today. ATP, okay, adenosine triphosphate, adenosine is one of our nucleic acids, right, is a currency. And for the most part, really one of the only currencies by which your cell functions. So when your cell wants to move things or do things, or we're going to make those transport vesicles move like gas, it all costs ATP. Okay? So ATP is a very important thing. Okay? Outside of regular functioning, it's also the not death thing that a cell needs to live. So making energy and making it well is very important. Mitochondria are very good at it. Okay, so most all cells have mitochondria. Okay, chloroplasts are also a type of organelle that do this. Okay, these are much more limited. Okay, they're doing, of course, photosynthesis, so this is only going to be found in our plants. Okay, so they have a special way to make currency. Okay, with the ability to use a different type of nutrient-rich system, specifically converting sunlight into ATP. Sounds pretty ritzy. Okay, so a plant would do both, right? They will have both chloroplasts and mitochondria. Go them. Okay, somebody like you and I only have mitochondria. Very disappointing. Okay, so we're going to go into the details of how they do what they do in a hot second. The last one I want to mention are peroxisomes. Okay, so these are special little sacs that you can find in cells. These are pretty common. Most cells have some of these, okay, but they're going to have very few, whereas a cell would be littered but have many mitochondria, they would only have a few, right? A small handful of peroxisomes. Okay. Now, as the name implies, okay, we see peroxide is part of the name. Okay, and that's exactly how it does its job. Okay, so these little sacs use hydrogen peroxide, okay, and as part of a chemical equation or a chemical reaction, take that hydrogen peroxide, okay, and with waste that they have inside, okay, change that hydrogen peroxide to water. Okay, you've probably seen this in action if you've ever been hurt and poured hydrogen peroxide on a wound where it kind of bubbles, right? So that's oxidation, that bubbly action. It's a high energy reaction, all that exciting bubbling. And then part of the liquid that you're left over with is water. Now, I'm not saying that you should do anything with what you're left with, because you do not convert all of that hydrogen peroxide to water when you do that on your wounds. Okay, but that is part of what's happening when you're doing that. That process that you're doing, though, creating that bubbling, releases energy. That's that bubbling action. Okay? These sacs in your cells do a lot of that, and they capture that energy that's being released, that ATP. Okay, now just like when you're doing this on your little wounds, okay, not always the most effective system. It does the job, okay, but we both know if we get seriously hurt, maybe hydrogen peroxide is not the way to fix it, okay? Just like if we need serious energy, a peroxisome is not the best way to do it. Okay, but it does work. It will give us energy, and it is helpful. Any questions about these? Okay, let's finish by talking about everybody's favorite organelle. Okay, the mitochondria, the mighty mitochondria, the powerhouse of the cell. Okay, so there's a reason it's considered the powerhouse of the cell, right? Remember, pretty much all cells have these, at least all eukaryotic cells have them, right? And the reason they're so good at their jobs is because they're efficient. And they're efficient because they have surface area. Okay, and this is a big theme on what makes any cell good at its job. Now, their surface area come from what's called cristae. Okay, so remember in our Golgi body, we had lots of folds called cisternae. Now, I don't name these things. Okay, and the mitochondria, they're called cristae. So we can look down at our image here and see all of these membrane folds. Okay, having all these cristae is like having a really good kitchen. Right? We all can picture what a really good kitchen is, right? Open with lots and lots and lots of countertops, right? The more countertops you have, the more cookies you can bake, right? You have more baking space, more space to lay the cookies, to cool them. Okay, you're a more efficient baker, the more countertops you have to work with. It's the same thing here, okay? The cristae are my countertops. So the more counters I have, okay, the more space I have to do my job. And in this case, my job is to make ATP. So I have more cristae, more countertops with which to make ATP. So mitochondria are very, very effective because they have lots and lots of space, lots and lots of surface area with which to do this. So that's what we mean by a large surface area, large countertop space to be very effective. And that's it. We're going to stop there. This will be the last thing that will be on your exam.